Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello, dear students. A very warm welcome to the TV learning programs. As usual, I'm Teacher Herbert, and I will be with Year 3 class, biology class, or Form 3. So we're still on Unit 20, which is genetics, the study of inheritance and variation. We talked about very many words about the study of inheritance and variation. We talked about how to determine the F1 generation using the Punnett square, and also using what we call genetic mapping. So today we're going to have lesson 5, which is haemophilia and even color blindness. These are genetic diseases or disorders that can be inherited. So the lesson has two main learning objectives. At the end of this lesson, you are able to describe and explain haemophilia and color blindness. And we also be able to show the relationship between genotypes and inheritance of haemophilia. So students, I hope you're ready with a pen and even a notebook so that you can take the notes and also make the lesson assessment. Bravo if you are ready with a pen and even with a notebook. Let's start the lesson. You know, most of the time when I'm going to start the lesson, I would bring some diagrams for you to understand. You remember we talked about some of the hormones, sex hormones that determine the sex, father characteristics and even mother characteristics, or female characteristics and male characteristics. We have these diagrams, as you see, when the father has haemophilia and the mother is unaffected. This is the father is hemophilic and the mother is unaffected. So meaning that is free from haemophilia. All right? What happens? These are what you call sex hormone that determines the sex. This is X, X and Y, X, X and Y. We are going to have X, XY. We're going to have X, XX. We're going to have X and Y. We're going to have X, XX. This is a baby boy who is unaffected. This is a baby girl, is normal but carrier. This is a baby boy, is normal. A baby girl is normal but carrier. So none of the sons, I'm talking about the boys, will have haemophilia. All of the daughters will carry the haemophilia gene. So the female ones, if the father is haemophilia, more chances of the daughters that are going to be produced are going to be hemophilic, which is also opposite. When the mother is hemophilic, most of the sons will be more affected compared to the daughters. When a mother carries the hemophilic genes and the father is unaffected. Now, see what will happen. Now, see, this is the father is not hemophilic, but the mother is hemophilic. What happens, see, the diagrams have, this is a boy, which is affected. This is a girl, okay, who is also affected. The boy is not affected, sure, but the girl is affected. The boy is, you see the boy is not affected, but the girl is also affected. So it means that there is 50 chances of each that will have the hemophilic. So it means that when the mother is affected, 50% of the boys can be affected. And also 50% of the girls can also be affected. Let's see on color blindness. That is color blindness. Color blindness. Most of the time we talk about color blindness test. Okay, that is confusion of the color. You see some that will be affected and others will not be affected. So let's start the lesson by talking about haemophilia. What is haemophilia? Haemophilia is also known as the breeder's disease. This is a condition of breeding without stopping. And we mentioned that this haemophilia is a sex-linked trait. When I talk about sex-linked trait, it is linked with sex chromosomes. Where the normal allele for the blood clotting is replaced by a defective recessive allele. The allele is located on the X chromosomes, not on Y chromosome. Students, remember I said Y chromosome does not carry any defect, because it is referred to as genetically empty. Therefore, the condition occurs more in males than in females. All right? Let's see. The clotting of the blood after injury is influenced by many factors. What are those factors that influence the clotting of the blood? This includes a protein known as what you call blood clotting factor, the platelets, simple. Platelets, which is a blood clotting factor 8, which is responsible to make the blood clotting. You remember I talked about how blood clots. Once you get injured, there's what you call triggering of the platelets to be produced, which comes to make what you call like a seal, and eventually stops the blood from clotting. We talked about the thrombokinase and the prothrombin and even the thromboplastin, which makes the blood to clot. So the inheritance of this factor is determined by two alleles. You remember when you talk about allele, I talked about two versions of the same gene is referred to as allele. So one allele determines the production of a normal clotting factor. This allele is referred to as a dominant allele. You remember when I'm using these terminologies, languages that I'm using to explain about genetic terms. These are the language that we use in genetics. Talk about a dominant allele. It's an allele that expresses itself in a phenotype. Okay? So this allele is a dominant that makes the blood to clot. The other allele influences the formation of defective clotting factor. This is also referred as a recessive allele. So the recessive allele is an allele that influences or is a defective that affects the clotting factor. So this allele that affects the clotting factor in biology are referred to as allele, and are referred to as a recessive allele. We have a dominant allele, we have a recessive allele. If you remember properly, I mentioned that recessive allele are written in small letters, then a dominant allele are represented by the capital letters. All right? So when a normal clotting factor is present, the blood clots normally after the injury. When you have a normal blood clotting factor, that is to say, when it is dominant. When you get injured, when you get damaged, easier to the blood to clot. When the defective clotting factor is present, the blood is thrown to clot or does not clot at all. Okay? The dominant allele is expressed with a capital H, where the recessive allele is expressed with a small letter. So if I say a dominant allele for blood clotting, I can use a capital H, that is a dominant allele. Okay? When I have HH, this means is a homozygous dominant allele, and that one can be having what you call a clotting factor. But if I have H and H small, normal but carrier in biology. Normal but carrier. This is a normal, but with a recessive gene, which is called normal but carrier. All right? We have H small and H small. Homozygous recessive. They are identical. And here it is very easy to express itself in phenotype student. When it is homozygous recessive, it means that character of a clotting factor, it is easier to be expressed in the phenotype. It can't express itself when it is in a heterozygous state. It can't express itself when it is in a dominant allele. All right? Let's have the diagram now. These alleles are located on the impaired part of the chromosome. Impaired part of a chromosome. Okay? Let me say, I will just try to use the diagram. Let me talk about possible genotypes. Possible genotypes. These possible genotypes for the normal, because let me have a wife, a mother. You know that the chromosomes we have X and X. But these X chromosomes, they can have what we call a superscript. This superscript can be either determining the normal because we said we have some characters, color blindness, hemophilia. It can just be located on what you call X chromosome, what you call sex chromosomes. They are linked on sex chromosomes. If I have the possible genotype here, I can have X with H capital and H capital. That is a normal female or a normal mother. If I have X, then I have H capital and H small. That woman is normal but carrier. Phenotypically, you can't understand that is affected. Okay? Then I will have X, all right? Then I have H small and H small. This woman is totally hemophilia. Once he gets damaged, it's hard for the blood to clot. Okay? So that is, we're talking about the female has genotype. It has the following genotype. This the female has the following genotype. These are the following genotypes I'm talking about. We have a normal woman. We have a normal but carrier. We have abnormal or affected woman. So the female has the genotype of X. This one is number 1. We have X, H capital superscript, X superscript, H small. So the normal clotting factor is produced in that way. So such a person is phenotypically normal females. But if the female has a genotype, this one, if they take a note of that, if a female has a certain genotype of X and H small, if he has that genotype, okay? The defective of clotting factor is produced. The blood is not clot and does not clot at all. Here he can breathe to the extent of dying. It means that he has that hemophilia. Okay? He is a homozygous recessive hemophilic. So such a person is phenotypically hemophilic female. That is a hemophilic female. Hemophilic female. Let me just use the word hemophilic female. This one is normal but carrier. This one is hemophilic. All right? So in males, the possible genotypes are, we have 2 possible genotypes. That's for the case of the male ones. We have 2 males. The possible genotypes are, we have X and Y. You know, these are chromosomes. These are sex chromosomes that determines the male. So it means that the possible genotypes I will be having we have X, H capital, and Y. This man is normal. All right? If I have X, H small, and Y, this one, let me try to underline. Take a note of that. This one is a hemophilic male. This is a hemophilic male. If that male gets damaged, if he gets injured, he can bleed to death. So you can ask a question to yourself. I told you Y does not have any superscript because Y is referred in biology is genetically empty. It doesn't carry any sex-linked character or traits. Okay? Let's go through the diagram. The relationship between genotypes and inheritance. Okay? We have the diagram. We have the chart. If you go through the chart. Okay? Let's go through the chart. I have a table. and you go through the table. Okay? There are 3. Let me try to allocate it here so that you can understand what I'm doing with you. You try to follow me and see what I'm doing with you. We have a table. The table has 3 columns: possible phenotypes, clotting factor, and even phenotypes. Is that even going there? Let me use that chart and we are able to understand it. We have X. This is a female. Possible phenotype. Then the clotting factor. If it has X and these are chromosomes that are located on sex chromosomes. That woman is normal. So that is normal. So phenotypically, once the woman gets damaged, this is a normal female, the blood can clot very easily. I have another woman which is here, which has got these allele. You have X, superscript H capital, and even H small. She is normal. Normal phenotypically. But genotypically, she is not normal because she has a carrier. I have that one, which is another woman, which is X, H small, and H small, because I had it here. This is a defective. She has a disorder, she is a hemophilic. Once she gets damaged, she can bleed without clotting. Some people, once they get damaged, they can bleed to the extent of death. So it means that they must be having some sex chromosomes, and they must be having what you call some of the chromosomes that are linked on sex chromosomes, which can cause the blood to clot without fading, which can make the blood to clot, which can make blood to fade to clot, and eventually you can bleed to death. I have a man who is X and H capital and Y. This man is normal, or this boy is normal. This one is a normal male or a normal father. But when you have X and H small here, you have a defective, you have a disorder of bleeding without clotting. And all these disorders, hemophilic, these are the conditions where a person can inherit from the father, because we can make a possible genotype, so we're going to do it maybe next time. Let me talk about maybe a normal man married, a normal man married an affected woman. What are the possible offspring that will be produced? All right? Let me just go through that. This is a normal man, a normal male, was crossed to an affected, to affected woman, affected female. I'm going to use the genetic mapping. Let me just use this supposed to be normal man, supposed to be X, and because it's not homophilic, with H small and Y. Then for the case of the affected woman, the woman is affected, is homophilic. Once is damaged, he can bleed to death. She is X, H small, and X, H small. Students, are you following me? Let's go through now for the formation of the gametes. I have X, H capital, and Y. These are, you have to circle the gametes for the factor of life. And here, this woman is a hemophilic. She can bleed without stopping. Get me very well, okay? We're going to have X, H small, and X, H small. Circle the gametes for the factor of life. We want to see the offspring that are going to be produced. Are the offspring going to be affected or not affected? Umugabo ntabwo afite indwara yo kuva, ariko umugore yari afite indwara yo kujya gute yo gukomereka akava till to death. Now, this will get a half of this and also gets a half of that. This will get a half of this and also gets a half of that. Let's go through. And you look for the diagram that we're going to get. Very easy. This is X, H capital, okay, and X and H small. Do you think that this is a girl produced? Is she normal? She's normal phenotypically, but has a carrier. There's what you call hidden character. Okay? We have this one. This is X, H capital, and yes, this is X and H capital, and X, H small. These are the girls. They are normal but carriers. We go here. This one and this one. This is supposed to be X and H small and Y. Okay? We have go here. This is X, H small, and Y. My friend, let's take a note of that, which is very clear. Let's take a note, seriously, and you understand this. I'm going to underline this. These are girls. These are boys. All the boys that were produced were homophilic. So once the mother is affected, more chances of affecting the sons is almost 50%. We have seen once the father is affected, more chances of affecting the daughters is also equivalent to 50%. So calculate now. This boy is homophilic. This boy is homophilic. So it means that 2 of the boys that were produced, 2 of the sons that were produced, it is 2 over 4 times 100, which is equal to 50%. Boys were homophilic. So if a woman is affected, almost 50% of the chances of the boys will be affected. When the man is affected, 50% of the daughters will be affected. So it is very easy. So these girls, they are not expressing it phenotypically. But once he gets a chance of getting married to a man who is also homophilic, automatically it will be expressed. So these are some of the diseases that can be inherited during the genetics. All right? This is very clear. Note that once a woman is affected, most of the boys are going to be affected. Good. So the heterozygous female, this is a heterozygous female. Here, if you see here, this is a heterozygous female. This is a homozygous female. This is homozygous. So is also referred to as carriers. This is a carrier. If I mention about a carrier, that is a hidden character. You can't see it phenotypically, outward appearance. But internally, this is what you call a disease which you can't identify it. So this is because she has a recessive allele for homophilic, which does not show in the phenotype. In the case when the homophilic male marries a normal female, all the girls will be carriers. Get the point? But where the boys will be normal. See the diagram now. This is a man, all right? See here. The female is normal. The male is normal. But the male is affected. The male is affected. Okay? But the female is not affected. The boys, all the girls, they are normal but carriers. But all the boys are normal. We have seen that one. 50% of the boy produced, when the father is affected, will be normal. You can also do it, which is very simple. And you try to see and you identify very clearly. Okay? Because if now, let me just try to show you, which is also very clear. Let me just use that side. Okay? If I have a man is affected, which is X and H small and Y. He got married to a normal man, to a normal woman. The woman is unaffected. You cross with a woman who is affected, who is not affected. This is X and H capital and X, H capital. There is formation of gametes. This is X, H capital. This is X, H capital. Circle the gametes as for the fact of life, or gametes has to be circled. So this is X, H small, and Y. Don't ask me a superscript on Y, because we said Y in biology is genetically empty. Does not carry any linked genes. There's no any linked trait on Y chromosome, because it's also very short. Circle. So if you draw it very clearly, draw it very clearly, draw it very clearly, okay? You draw it very clearly. I will be having, this is X, this is H capital and H small. I will have X, H capital and XX, okay? We have X, yes, I have, for the case I have X, H capital and Y. I have X, H capital and Y. My friend, note that. The girls here, when the man is affected, the girls will be normal but carriers. There is what you call a hidden character. You can't see it, but you can see it in a genotype. But all the boys that were produced, 50% of the boys produced were normal, which is very clear. So color blindness Let's go to what we call color blindness now. Color vision is determined by a pair of genes. The genes for the normal color vision is dominant because to see clearly, that is to say, you are able to identify the color. So a normal version is a situation whereby the individual is able to distinguish all colors of a given subject. So the genes can also be abnormal color visions, or vision is recessive, of course. It brings about a condition known as what you call confusion of color, green and red color blindness. So this is inability of given individuals to distinguish a red from green color. Okay? Which is clear. Okay? So the allele for the color vision is also linked to the sex, is also a sex linked. They are found on the X chromosomes and they are absent on Y chromosomes. Okay? So if a mother has a defective, 50% of the chances of the son will have the defect. But when the father is also having a defect, almost 50% of the daughters produced will be carriers. Okay, which is here. You go through the diagrams, you are able to understand it. They say what you call color blindness, all right? So that is the diagram, and you see when you have that diagram, this is the diagram is supposed to be a normal female. This is a normal but carrier female. This is an affected female, which is color blindness. We have this one, which is a normal male. We have an affected female, which is color blindness. If you cross that one, you also go to the same results as I did it in the previous lesson. Okay? So this is if a man with a normal color vision marries a color blindness woman, and they have the children, then the possible genotypes are here. You have to go through the genotypes. So student, we are coming to the end of the lesson, but I can't leave you without testing your understanding. So I'm going to leave you with a lesson assessment. There are almost 6 numbers. What is haemophilia? On which chromosomes is genes are created that causes haemophilia linked to? So we have the number last, which is very good. A woman who is a carrier for haemophilia marries a man who is hemophilic. Could any of their children have hemophilic? If so, would the children be married or be male or female? So student, thank you very much for your active participation and listening. Enjoy the rest of the day and stay safe. See you next time.